Welcome to this explainer, everyone. Today, we're taking a grand tour of the most fascinating, bustling, and literally the most densely packed city in the known universe. But, you're not going to find this city on any map. You'd have to hunt for it under a microscope. We're talking about the cell. Far from just a simple, static building block, the cell is this fully functioning, microscopic metropolis. It's got its own power plants, factory lines, security forces, and central command centers. It's the structural and functional unit that literally built you, and it works tirelessly 24/7 just to keep you alive.
Okay, let's dive into our roadmap for today. First up, the building blocks of life. 2, the prokaryotic world. 3, eukaryotes, plants versus animals. 4, the cell factory system. And finally, 5, powerhouses and control centers. Let's get into it.
So, starting off with Section 1, the building blocks of life.
To really grasp this core unit, we've got to look at how it was even discovered. Think about it. Whether you're a single-celled organism operating totally solo or a complex human with trillions of specialized cells dividing up the labor, every single metabolic reaction keeping you alive happens right inside the semi-fluid substance of the cell, the cytoplasm. And figuring out how this whole thing propagates, well, that brings us to a phrase that honestly sounds a bit like a magic spell, omnis cellula acellula. Coined by Rudolf Virchow back in 1855, this famous Latin phrase simply means all cells come from pre-existing cells. That was it. That was the brilliant final missing piece of the cell theory puzzle, perfectly explaining how life actually propagates and grows through cell division.
Let's quickly trace how rapidly our understanding of this metropolis evolved. It all kicked off in 1665 when Robert Hooke discovered the very first cell. Well, a dead cork cell, but still. Then things really heated up in the 1830s. A German botanist named Schleiden realized, hey, all plants are made of cells. And right around the same time, a zoologist named Schwann realized the exact same thing for animals. The two of them collaborated to draft the initial cell theory, which Virchow then finalized. Together, their work proved, without a shadow of a doubt, that absolutely everything living is a product of these microscopic cities.
Moving right along to Section 2, the prokaryotic world.
Let's travel back in biological time to the primitive, older district of our cellular city. The word prokaryote literally means before the nucleus. So in these organisms, like bacteria in kingdom Monera, their genetic blueprints are just lying out in the open in the cytoplasm, totally naked, no protective nuclear envelope around them at all. But despite that internal simplicity, just look at their incredibly complex exterior defenses. The outermost layer is the glycocalyx, which can be this tough capsule or a loose slime layer. It basically acts like a cloaking device, helping the bacteria completely dodge the host's immune system. Beneath that, you've got the rigid cell wall, acting like the physical city wall to keep the cell from bursting. And finally, the cell membrane, which is the security gate determining exactly what gets to enter and exit. Now, since these prokaryotes don't actually have all those complex membrane-bound organelles inside, they have to get a little creative. They do this by literally folding their security gate, the plasma membrane, inwards. We call these clever little infoldings mesosomes. By drastically increasing the surface area, mesosomes step up to handle the heavy lifting, complex jobs like cellular respiration, and even the distribution of DNA to daughter cells. Honestly, it's just a perfect example of making the absolute most out of extremely limited cellular infrastructure.
Now for Section 3, eukaryotes, plants versus animals. Unlike those older districts, eukaryotes have a true, well-defined nucleus and are highly compartmentalized. They have distinct organelles, all separated by membranes. It's a modern, sprawling metropolis. But within this eukaryotic world, we actually see two very different architectural styles, plant cells and animal cells. The absolutely crucial point here is knowing how to tell them apart. And the best way? Just remember the 4 C's mnemonic. Plant cells have a rigid cell wall, food-producing chloroplasts, and a massive central vacuole for storage, but they lack centrioles. Animal cells are the exact opposite. They possess centrioles for cell division, but they totally lack those other 3 features. Just keep those 4 C's in mind, and honestly, you'll never confuse the two again. To understand how the borders of these modern cities operate, we have to look at the Singer and Nicholson fluid mosaic model. Think about it. It's not a solid, rigid wall. It's actually a dynamic, fluid sea of lipids, and floating right in this fluid lipid sea is a mosaic of proteins that act as carrier tunnels for molecules to pass through. Oh, and attached to the outside? Carbohydrates. They basically act like biochemical shipping tags, so different cells can actually recognize each other. And that brings us to Section 4, the cell factory system. Stepping inside this highly interconnected industrial district, you'll see it operates exactly like an automated factory line. These organelles work incredibly closely together to make sure the cell produces, packages, and ships exactly what your body needs to survive. First up, the rough endoplasmic reticulum, which is studded with ribosomes, manufactures proteins. Meanwhile, the smooth ER handles synthesizing lipids and steroids. Then, all these raw products get shipped straight over to the Golgi apparatus. The Golgi modifies them, literally applying those carbohydrate shipping tags we just talked about, and then packages them into vesicles. This ensures every single product is delivered to the exact right address, whether that's inside or outside the cell.
But what happens to the waste in this industrial district? Check out this number, 4.5. That is the highly acidic pH maintained inside lysosomes, often called the suicide bags of the cell. Lysosomes use specialized proton pumps to just constantly drag in protons, maintaining this insanely intense acidity. Why? Because the powerful hydrolytic enzymes inside them literally require a pH of 4.5 to activate. This allows them to effectively break down and completely dissolve any cellular waste or debris.
Let's move to our final section. Section 5, Powerhouses and Control Centers.
So we've seen the walls and the factories, but how is all this machinery actually powered? And where are the architectural blueprints kept? Well, eukaryotic cells rely on mitochondria, the literal powerhouses that generate your ATP, energy currency. And, in the case of plants, chloroplasts, which use light to produce food. But here's what's truly mind-blowing. These organelles are semi-autonomous. That means they actually have their own circular DNA and their own ribosomes. They operate almost like independent foreign factories, just living inside the cell. Speaking of ribosomes, let's highlight these vital little protein factories, discovered by George Palade back in 1953. Because they don't have a membrane, they are universal. You'll find them in both prokaryotes and eukaryotes. But let's do some quick biological math here, okay? A prokaryotic ribosome is 70S, and it's made of a 50S and a 30S subunit. Wait a second. 50 plus 30 definitely equals 80, not 70. The math doesn't seem to add up at all, right? Well, that's because the S stands for sedimentation coefficient. It actually measures how fast they drag through a fluid under gravity, not their direct physical weight. It's just a fun little biological quirk.
And you know what? This quirky structural math applies to the cytoskeleton too. When we look at structures that move the cell around, like cilia and flagella, they have a 9+2 arrangement of microtubules. That's 9 doublets on the outside and 2 singlets right in the center. But the centrosome, which anchors the cell during division, that has a 9+0 cartwheel structure. 9 triplets around the edge and absolutely 0 in the center hub. Honestly, it is microscopic engineering at its finest. Finally, we reach the nucleus, the central command. Inside here, long strands of chromatin pack themselves down tightly into chromosomes, which are essentially the ultimate DNA storage vaults preparing for cell division. And their distinct shapes? Those are entirely determined by the location of the centromere that binds them together, from the perfectly centered metacentric chromosome all the way to the extreme tip of the telocentric chromosome. This incredibly precise packing ensures that absolutely no genetic data is lost.
Which leaves us with this final, staggering question to ponder. How does a single microscopic entity, packed with mere chemical blueprints, know exactly how to build, maintain, and perfectly duplicate an entire human body? We've explored the layout of this microscopic metropolis today, but how this city divides and replicates itself without making catastrophic errors, that is a true biological miracle. And that's exactly what we're going to unpack next time. Thanks so much for joining me on this explainer. I'll see you in the next one.